Hello students, this is Jyoti Rajput and today's topic is distinguishing characters between the prokaryotic cell and eukaryotic cell. So in this topic here we are going to distinguish between the prokaryotic cell and the eukaryotic cell. So the very first characteristic is the size of the cell. Prokaryotic cell, the size is typically ranging from zero point 0.2 to 2 micrometer in diameter. But when we talk about the eukaryotic cell, the size is about 10 to 100 micrometer in diameter. Next is about the nucleus. Nucleus, it is absent in prokaryotic cell. But when we talk about the nucleus, if it is present, then it is of a primitive type, which we called as nucleoid. Nucleus, it is present in eukaryotic cell, so that is why we call them. U means true and karyot means nucleus. So true nucleus is present in the eukaryotic cell where there is presence of the nuclear membrane and nucleoli is present. Next is membrane enclosed organelles. Membrane enclosed organelles, they are absent in prokaryotic cell, but here they are present in the eukaryotic cell. When we talk about the lysosome, Golgi complex, endoplasmic reticulum, all these are organelles which are made up of single membrane, which are having single membrane. When we talk about mitochondria and chloroplast, they are having double membrane. So these are double membrane bound organelles. Next is the flagella. In the prokaryote, the flagellate consists of two protein building blocks. But when we talk about the eukaryotes, the flagellate is complex and it consists of multiple microtubules. Next is about the glycocalyx. Glycocalyx, it is the outermost membrane. So here it is present as a capsule or a slime layer in the prokaryotes. And in the eukaryotes, the glycocalyx, it is present in some cells that lack a cell wall. Next about the cell wall. Cell wall, it is present in the bacterias, that is called as peptidoglycan. And when we talk about the archaebacteria, they have the pseudomurine layer. Pseudomurine, it is present. Here the pseudomurine is here. This is how the cell wall is made up of pseudomurine in the archaebacteria. As we all know, the archaebacteria, these are the bacteria which are present in the adverse environmental condition, like in high temperature environment, very cold temperature environment. So in the adverse condition, the bacteria which is present are the archaebacteria. So the cell wall, it is made up of pseudomurine. But when we talk about the normal bacteria, the cell wall, it is made up of peptidoglycan. Here, the cell wall in the eukaryotes, if it is present, then chemically it is very simple. So cell wall, it is present in plant, cell wall, it is present in fungi, but in animals, the cell wall, it is absent. Next is about the plasma membrane. So in prokaryotes, no carbohydrate is present in plasma membrane, generally lacks sterols. And in eukaryotes, the plasma membrane, it has sterols and carbohydrate, which act as a receptors. Next is cytoplasm. In the cytoplasm in prokaryotic cell, there is no cytoskeleton or cytoplasmic streaming, but it means cytoplasmic streaming, it is less. Next, here in the eukaryotes, in the cytoplasm, there is presence of cytoskeleton like the microfilament, microtubules, intermediate filament, and cytoplasmic streaming do occur in eukaryotes. Ribosome. In the prokaryotes, the ribosome is of 70s type, and in eukaryotes, the ribosome is of 80s type. But when we talk about the organelles like the chloroplast and mitochondria, the ribosome inside is of 70s type, like that of the bacterial origin, like that of the prokaryotic origin. Chromosomes. In prokaryotes, usually there are single chromosome present, single circular chromosome are present, but they don't have histone. Histone, it is always present in the eukaryotic cell. It means with the DNA, the histone are present. So there are multiple number of chromosomes, multiple linear chromosomes are present, and chromosomes are always associated with the histone protein. So histone, they are always found with the eukaryotic cell. Cell division. In the prokaryotes, usually the binary fission type of cell division is present. And in eukaryotes, there is mitosis type of cell division is present. Next is sexual reproduction. There is no meiosis seen in the prokaryotes. Meiosis, where there is reductional division of chromosomes. So no meiosis are seen in the prokaryotes. There is transfer of DNA fragments. It is only and only by the conjugation. And in यूकैरियोट्स it involves the meiosis. ऐसा नहीं है कि माइटोसिस नहीं होता है, माइटोसिस भी होता है, लेकिन सेक्सुअल रिप्रोडक्शन के दौरान जो है there is presence of the meiosis in the eukaryotes. Here there are some example of the prokaryotes like the bacteria, the archaebacteria, and here the example of eukaryotes are plants and animals. And the prokaryotes, they were the very first who appeared. So first they appeared about 3.5 by 10 raised to 9 years ago, and the eukaryotes they arrived later, so they appeared later. So it is about 1.5 by into 10 raised to 9 years ago. So here we are going to discuss about the chapter Introduction to Cell Biology questions. So these are the questions which are asked about this chapter, like the definition, or you can say explain cell wall, cytoplasm, prokaryotic cell, lysosome, eukaryotic cell. And there are some short answers being asked, like what is the structure of eukaryotic cell, draw the typical diagram of plant cell, draw the typical animal cell, draw the structure of the typical E. coli cell, sketch and label prokaryotic cell, describe the structure of eukaryotic cell. Some more questions which are asked on this chapter is differentiate between prokaryotic and eukaryotic cell, explain the structure of E. coli with labeled diagram, explain the structure of plant cell with labeled diagram. So this is how the chapter ends, students. Uh, and the chapter was Introduction to Cell Biology. So this is how the video also ends, and thank you very much.